Good day, grade 12 learners. Today we are going to study life sciences, but a specific topic called scientific investigations. We are collaborating from Johannesburg West. My name is Fawzie Rahiman, the facilitator for life sciences from Johannesburg West. I'm collaborating with ThunderEDUC, and we are trying to bring you the topics that are of concern in the final exams. Many of you are having difficulty with scientific investigations, so we are going to try and iron out some of the creases. So why must we do scientific investigations? Scientific investigations fall under specific aim 2, which requires us to doing science, practical work, and investigations. So that is why we have to be involved in scientific investigations. Now, what is specific aim 2? Specific aim 2 is investigating phenomena in life sciences. So you must be able to carry out investigations, solve problems that require practical ability. The ability is underpinned by the attitude of curiosity and interest in wanting to find out how the natural world and living things work. So we are related to what is happening in the environment, related to life sciences. Now, every test and every exam must have a question on scientific investigations, because the minimum of scientific investigations is approximately 20% of the total paper. So we're looking at approximately 15 marks in every paper that you write, every test, depending on how much the test is out of. So in your final exams, approximately 15 marks for paper 1, 15 marks for paper 2, that is 30 marks that is allocated to scientific investigations. And our learners are not performing well in this topic. So let's start. What does the diagnostic report say? The diagnostic report is a report that is written by that comes out at the end of the year after the marking process has been conducted. And we look at what are the high frequency errors and what teachers or learners can do to remediate that. So what are they saying? They're saying that a greater emphasis must be placed on practical work and practical tasks, and good quality grades in grade 10 and 11 will assist in preparing learners more adequately. That means you need to start doing this from grade 10. Learners must be challenged to design their own investigations by given an observation, hypothesis, or aim. And that is why you have done your practical investigations this year with regards to the genetics hypothesis testing. Remember, you worked out the ear, the hitchhiker's thumb, whatever it is, but you were given a task that you had to do a practical investigation. And that is part of learning the skill of scientific investigations to prepare you for paper 1 and paper 2 final exams. Now, what is the scientific method? The scientific method involves curiosity. Remember, we said curiosity. That is some kind of observation. You observe something and then you question, why is this happening? You come up with a hypothesis after observing. That is why we say the hypothesis is an educated guess or a smart guess. After you observe over a long period of time, you observe a situation, you come up with a hypothesis, and then you conduct an experiment, you analyze your results, and then you come up with a conclusion. If your conclusion doesn't speak to what is occurring, then you can always go back and, or not, doesn't make sense, in other words, you can always go back and start a new question. And then you continue with this process until you come up with something that is satisfactory. Now, the terminology you need to know. Remember, you start off with terminology in your exam guidelines. For every single topic that you engage in, you must look at terminology. So in the same way, for scientific investigations, there are terminology that you must know. First one, observation. What is an observation? Scientists saw, they hear, they encounter, and this encourages them to investigate further. Then we look at a hypothesis. It's a possible prediction or an explanation of the relationship between two variables. The aim, it usually starts off with words to investigate, to find out. And it includes both variables. It describes what the investigation is trying to find out. So the aim is always found at the beginning of the investigation, and it tells you what the investigation is trying to find out. So it has words, as we said, to investigate, to find out, to determine, okay? Those are the words that are usually present when we are working with the aim. Now we come to the variables. We're looking at the independent variable and the dependent variable. The independent variable is the one that the scientists control. So if I want to conduct an investigation and I want to say I want to have it over 10 days, I'll take the results from day 1, day 2, day 3, then I am controlling the days. So I am controlling the independent variable. It is manipulated by the person who's doing the experiment, the researcher, the person who's doing the experiment. The dependent variable responds to the independent variable. Therefore, it's called the responding variable. It is usually the variable that is measured. So if I want to work out the height of the plant, how long is how much the plant is growing, how high the plant is growing, I decide I'm going to take the results every 10 days. So my days are going to be the independent variable. I'm choosing the days, but the dependent variable is going to be the height of the plant because I am measuring that. That depends on the day. The controlled variables, these are variables that must be kept constant to ensure that, so that the investigation is valid, so that any effect is due to the change in the independent variable only and not anything else affecting the investigation. And the control, it allows for a comparison with the results of the experiment. The control is identical to the experiment, except that the factor that you are testing is removed from the control. So in other words, the control is identical to the experiment, except that it excludes the variable that is being tested. So then we will remove that factor and then we will add it in the experiment, and then we can compare to say, without it, this is what happened. When we added this factor, this is now what happened, and we can use that then to compare. That is the control. Accuracy refers to the care taken when taking measurements, and validity. Validity speaks to experimental design or experimental method, how appropriate it is in addressing the aim of the investigation. For example, keeping all factors constant, identifying the controlled variables help in making the investigation valid. So for example, I want to give you an example here. If I am measuring the height of the plant, I must use plants grow tiny amounts. So I must use now a measuring tape, also not to damage the plant. If I use a big ruler, that's not going to give me an accurate result. And then if I use the measuring tape, I must continuously use the measuring tape, not change into using other apparatus to measure. That is validity. Results. The evidence produced during the investigation that will either support or refute the hypothesis. This may be presented as an extract. In our case for life sciences, most times it's a table, a graph, or a diagram. Now, let's look at reliability. The results can be made more reliable if the investigation is repeated, a bigger sample size is used, the samples are taken randomly, and many readings are taken to obtain an average. But remember, For validity and reliability, we don't want a generic answer. We don't want you to say, repeat the investigation, increase sample size. We are moving away from recipes that are producing generic answers. We want you to look at the investigation. See, how many times was this investigation conducted? Okay, it was conducted 3 times. Therefore, to improve reliability, I must repeat the investigation more than 3 times. If the sample size was 200 women, in order to improve reliability, I must use more than 200 women. But if the question asks you, is this result reliable? You can say yes, because 200 women were used and that is a large sample. So you see how you have to relate your answer to the investigation that is given to you. You no more can say increase the sample size, repeat the investigation. That is not accepted anymore. So we don't want a generic answer. We want to see if you understand what is reliability and validity, and therefore your answer has to be talking to the experiment, the question that you have in front of you. So it is critical that you read your question well, highlight, underline. Remember, the question paper is your question paper. You can circle all the numbers, highlight, underline, so that it makes it easy for you when you are trying to answer the question. But please, the step number 1 is to read the question carefully. The stem of the question is all the information that is given before the questions, and that is critical in understanding how to answer the questions. So you're going to read the question, you're going to look at the data and unpack the data. What data do I have? I have a table. Which is on the right-hand side, what is on the left-hand side, what is at the top, my columns and my rows, what is written there. So you have to do that in order to answer these questions well. So let's move on, and I will show you when we do examples. The conclusion is directly linked to the aim of the investigation, and it either confirms or refutes the hypothesis. Okay, and this will become more sensible once we do more activities. Right, so what is the difference between an experiment and an investigation? A scientific experiment is a procedure used to showcase scientific principles without following a scientific method. Okay, it's just an experiment that varies in your scientific investigation because we are hoping to prove a known scientific principle. We don't follow a set number of instructions. Now, the aim is to determine if light is needed for photosynthesis in an experiment. But in an investigation, it is a process. Okay, it's a process, not just a procedure. It's a process whereby a hypothesis is developed, data is gathered, an experiment, many experiments are conducted, data is analyzed, and conclusions are drawn. So the experiment itself is showcasing or doing the experiment, but an investigation is a whole process where many experiments are conducted. Scientists use scientific investigations to understand how the world works to solve problems, and this differs from an experiment because multiple variables are tested and experiments are repeated to gather data which can be used then to draw conclusions. So here we said to determine if light is required or needed for photosynthesis. Here we are saying the effect of light intensity on the rate of photosynthesis. So we will do many experiments and use various light intensities and then measure the rate of photosynthesis. Now, let's look at the different steps of the scientific investigation. Remember, we said you're going to observe, then you'll question, you'll come up with a hypothesis, you'll conduct various experiments, you'll come up with a conclusion, and then you'll come up with your results. But the first thing you've got to do is you've got to plan. So you need to look at any investigation that you're going to do, you need to plan first. And you need to look at the context of the investigation. Are you working with people? So if you're working with people, then you need to get permission from people. But if you're working with bacteria, for example, you don't need permission to work with bacteria, right? You don't need to ask their permission to do research on them, but you need to ask people if you are working with people. Now, the key word for planning is the word decide. If you have the word decide in your planning steps, then it means that you are planning. You are not doing. You are not conducting the investigation. So if you have the word decide, then we know when we are marking your papers to say this person is planning, because they are still deciding. They're not conducting. So that word decide is the key word when you are writing your steps or writing your planning. The first thing you got to do is decide on a time, a date, and a place to conduct the investigation. Then you need to decide what apparatus I need, what materials need to be used. Decide on how I will record all the data. Decide on how many participants will I use, right? The number of participants, if people are used, or the sample size, if you are using other organisms like chickens, or if you are using apparatus like test tubes, what will my sample size be? Okay. And remember, the word decide is for decide on a time and a place. Decide on the apparatus. Decide on how the data will be collected. Decide on the number of participants. Decide on which factors will be kept constant. Decide on the composition of the sample. Okay, decide on indemnity forms for participants if you are working with humans, if you are working with your fellow students, if you are working with people at a hospital, people at a school, people at a church. And then how will you recruit? You will decide again on how will you recruit participants, or how will you get volunteers to participate. But the word decide here, as I said, is the key word that must be used when you are planning. Now we're going to look at hypothesis. The hypothesis is a prediction. Remember, we said it's a smart guess. It must be testable. It can't be anything. It must be testable. And there must be, the way you write it, there must be a relationship between two variables. Okay? So there's always two variables, the independent variable and the dependent variable. It's important to note the hypothesis can never be a question. It should be testable, and if it could be an incorrect relationship because it still needs to be tested. So if you write something that is not right, doesn't matter. When you test it, you can say, then I do not accept my hypothesis. The hypothesis is allocated 2 marks. So let's look at an example. Feeding chickens yellow grains will increase their rate of growth. So what are you doing? You are feeding them yellow grains. That's the independent variable. You are manipulating that variable. You are giving them yellow grains. But the relationship is, you are saying it will increase their rate of growth. You're going to measure the rate of growth. That is your dependent variable. So you could say feeding chickens yellow grains will decrease their rate of growth. Then you will conduct the investigation and you'll say, oh, you know what? It actually increases their rate of growth. Then you will not accept that hypothesis, because the hypothesis is it will increase. And therefore you will do the experiment again and say yellow grains increase the rate of growth. And therefore then you will conduct the investigation and you will say, I accept my hypothesis. So that's how you would write a hypothesis. Now, we are looking at the variables. The first one is the independent variable. It's a factor that is being investigated to investigate whether caffeine affects stamina. Remember, the factor is usually manipulated or changed by the investigator or the researcher, either at the start of or during the course of the investigation. When you are drawing your graph, the independent variable always occurs in the x-axis. So you're not going to write here independent variable. No, you're going to write what the independent variable is. So you're going to say caffeine. This is going to be marked right, okay? Not this. Then, this is your x-axis. This is just to show you. Now, your dependent variable. Your dependent variable is a factor caused by the effect of the independent variable, and it can be measured. So, to investigate whether caffeine affects stamina, stamina will be your dependent variable in the investigation, and you can measure stamina. Okay, you can give a person caffeine, and how much caffeine you give them, how often you give them, the dose of the caffeine. That's you as the researcher, as the investigator is going to manipulate. And then you'll measure the stamina after that. So therefore, the stamina depends on the amount of caffeine. Therefore, stamina will be the dependent variable. The effect is usually measured in some way and appears on the y-axis. This is your y-axis, and here you'll say stamina. And remember, when you're writing that, you must have your units. So a dependent variable can be measured directly or indirectly, example by observation. Now, where do we get our variables from? This is very important, okay? You get your variables by looking at the aim of the investigation. That's why you need to know how the aim is written. Using the result instead of the aim to extract variables sometimes leads to incorrect dependent variables. So sometimes the results indicate how the dependent variable was measured. Let's look at this. This table here shows results of the investigation. It tells us the temperature in degrees Celsius and the average mass of the chicken. This tells us how they measured the growth of the chicken. They measured the growth of the chicken by measuring the average mass. So if we look at the aim is saying to determine the influence of temperature on the growth of chicken. So our independent variable is temperature, just temperature. And our dependent variable is growth of the chicken. But if you use the table, if you use the results, you would have got this wrong, because you would have said temperature, but average mass of chicken, you would have said average mass of chicken. And that is telling us how they measured the growth of the chickens. It's not telling us the dependent variable. So be very, very careful. Use the aim of the investigation. The second thing I want to highlight is that you cannot say to determine your independent variable is the influence of temperature. No, it's not the influence of temperature, because this could be a high amount or a low amount. We are looking purely at temperature on the growth of chickens. So the independent variable is just temperature, not the influence of temperature, because this implies that you have measured already and we are still conducting the investigation. And as I said, the dependent variable is the growth of the chicken. So therefore use the aim. Very important, use the aim and not the results. Not the results, because then you'll get the wrong dependent variable. Now, we want to look at the variables. Sifiso, so I'm doing an example. Sifiso wants to investigate the effect of different temperatures on the growth of grapevines. Remember, the effect we don't use that. So the independent variable will be different temperatures, and your dependent variable will be the growth of the grapevines, because you are manipulating the different temperatures and you are measuring the growth of the grapevines. So he designs an experiment to determine the weight of the grapes at varying temperatures for 90 days to allow for maturity. Okay, remember the exam question paper is yours. Circle all your numbers. He maintained a sufficient sunlight, well-drained soil. The table below shows the data on the mass of the grapes produced by the vines at the end of the investigation. Study the results and answer the questions that follow. Now, this is also a data response question where you are responding to data. You have been given temperature. So let's look at the table. My table has 2 columns. The heading on my one column is temperature. The other one is the mass of the grapes produced, and that is in kilograms. And this is telling me how the growth of the grapevine, of the grapes, were measured. And we got it by measuring its mass. Then I've got temperature in 10, 15, 20, 25 degrees Celsius, going up in an increment of 5. And my mass is 0.5, 1, 1.5, 2.5. So that is increasing here. But if I look at 10 degrees, it's 0.5, 15, 1, 20, 1.5, 25, 2.5. So you need to look at all of these factors on your table before you even look at the question. You can come up with a relationship. The temperature is increasing. My mass of my grapevine, of my grapes produced, is also increasing. So let us move on and remember that my dependent variable is the growth of the grapevine. How did I measure the growth? By measuring the mass of the grapes produced. So therefore, this cannot be your dependent variable. This is your dependent variable, and you get that wants to investigate. Therefore, this is my aim. Okay? Now, we're going to look at validity. What is validity? Validity speaks to experimental design. Anything to do with the experimental design will affect or improve the validity. Affect it negatively or positively. Let's see. All the factors that can influence the result must be kept constant, so that we know if we are investigating a certain aspect, that aspect alone must be giving us the results, not any other factor that can change the results. So you must be encouraged to use the term validity, and we don't use the term fairness. Please stay away from that. The validity of the investigation increases or is improved when all factors that can influence the investigation are kept constant, besides the one that you are changing. So besides the independent variable, remember, you are changing the independent variable. You are manipulating the independent variable. That's the only variable that can change. Everything else in the investigation must be kept constant. So we know that whatever the result is, it is due to only the variable that we are changing, the independent variable, not any other factor. Right. So let's look at this. Safiso wants to investigate the effect of different temperatures on the growth of grapevines. He designs an experiment to determine the weight of the vines at varying temperatures for 90 days, varying temperatures. So let's look at this firstly. We said that the aim is to investigate. It's telling us why we're doing the investigation. So that's our aim. And from the aim, we get our dependent and independent variables. We said the different temperatures is our independent variable, and the growth of the grapevine is our dependent variable. Now, let's read the stem of the question. This is all the information given to us before the investigation. He designs an experiment to determine the weight of the grapes at varying temperatures for 90 days to allow for maturity. What did he keep the same? He maintained sufficient sunlight and well-drained soil. Okay, so that was giving us the validity. The table shows the data on the mass of grapes produced by the vines at the end of the investigation. Study the results in the table and answer the questions that follow. Okay, so that's what you get. Let me just go back. So this is now looking at the validity. So we are only changing the temperature. Only the temperature can change, nothing else. And we will then work out the mass of the grapes produced. So this is what we are measuring. Therefore, this is what we are measuring, the mass of the grapes produced. And this is telling us the growth of the grapevines. And that's giving us our dependent variable. Now, he had sufficient sunlight and well-drained soil. So if he had waterlogged soil, or if he did not have sufficient sunlight, then he is changing the variables, and that is going to affect our results negatively and decrease validity. Right, we're now going to look at reliability. Sifiso wants to investigate the effect of different temperatures on the growth of grapevines. Again, that's our aim. He designs an experiment to determine the weight of the grapes at varying temperatures to allow at varying temperatures for 90 days. Now, he is not doing this experiment for 1 day. He's not doing it for 1 week. He's doing it for 90 days. That tells us that he conducted the experiment over a long period of time. Therefore, it is a reliable result. Remember, reliability. If I do the experiment in South Africa, for example, and somebody wants to conduct the same experiment in Japan, they must follow the same procedure and they will get a similar result, almost the same result. That means my result is reliable. So the more samples you use, the longer period of time you conduct the investigation, the closer you'll get to the true value. And that will then make your result reliable. Another example is if you are taking the average of the class. If you take 2 people, you're going to get a certain number. But if you take 40 people, you're going to get closer to the true result. Okay, so that's what reliability is. Now, he maintains sufficient sunlight and well-drained soil. That speaks to experimental design. That is validity. How long he did the experiment for and how many participants he used, that speaks to reliability. So you must know the difference. The control in an experiment is set up in exactly the same way as the experiment, but it excludes the variable that is being tested for. In this investigation, we want to see if carbon dioxide is given off during respiration. So we've got a setup here where we have a tube with fresh green leaves, which will undergo respiration. We have a bag of worms, which will undergo respiration. Then we have one here, C, which has nothing inside. Can you see? There's no worm, there's no leaf, there's just plain air. Our indicator to show us whether respiration has occurred will be, what would that be? It will be clear lime water. You must say clear lime water. What happens to clear lime water in the presence of carbon dioxide? Clear lime water turns milky. So what's going to happen? This clear lime water here in this test tube, they're going to turn milky in the presence of carbon dioxide because the leaves will respire and the bag of worm will respire. Now, we are testing for respiration where the CO2 is given off. So CO2 will be given off by the leaf, CO2 will be given off by the worms. Is there anything here? We are testing for CO2, therefore CO2 is removed from this test tube. We have plain air, right? Now, we are going to see if carbon dioxide is, and this air will not have carbon dioxide, okay? This air will not have carbon dioxide. So now we will look to see what happens. In this test tube A, this clear lime water turns milky. In test tube B, the clear lime water will turn milky. But here in C, the clear lime water will remain clear. Why? There's no CO2. That means there was nothing here to undergo cellular respiration. But here, not cellular respiration, sorry, respiration. Here, respiration has occurred and therefore carbon dioxide was given off. So can you see we can compare? We can say that the fresh green leaf underwent respiration and carbon dioxide was the gas that was given off because carbon dioxide turned the clear lime water, it made it milky. The bag of worms underwent respiration and gave off CO2, and the CO2 turned the clear lime water milky. But the air here was free from carbon dioxide did not turn this clear lime water milky because there was no respiration occurring, no CO2 was given off in this atmosphere here, and therefore this one was clear. So test tube C is the same as A and B. It has exactly the same size, the same amount of carbon dioxide indicator or lime water, the same type of seal at the top, the rubber seal at the top. Everything is the same. Besides, the factor you are testing for has been removed. I'm testing for CO2 if it's given off during respiration. I don't have anything in there that can cause CO2 to be given off, and therefore it shows me that whatever was it shows me that the leaf and the worm gave off CO2 because that turned the lime water milky. So A and B does not have a living organism that can respire, so the CO2 indicator did not change. CO2 indicator is compared to CO2 indicator in A and B, where respiration is taking place, and here there's no respiration taking place here. Therefore, there was no CO2 given off. Okay, I hope you understand the control. In every experiment, there must be a control. Okay, moving on. We are now going to look at a question on scientific investigations, and all the factors I have been explaining to you all along. You're now going to put it into use to see how we can answer our questions using what we have been taught already. Right. The first thing is, I said, you must read the stem of the question. Do not go to the questions without reading what the scientific investigation is all about. Secondly, you're going to unpack your data, look at the table, understand what is happening in the table before you move on. An investigation was carried out to study the effect of temperature on the action of pepsin. So this tells me that an investigation was carried out to study. This is what the aim of the investigation was. So the one in red is my aim. To study the effect of temperature on the action of pepsin, a protein digesting enzyme in stomach fluid. The investigation was set up as follows. 10 grams of protein was placed in each of the 5 test tubes. In other words, I had 5 test tubes. Draw on your question paper if you have to. Remember, it's your question paper, guys. You need to draw on your question paper, write, circle, underline, highlight on your question paper. So 5 test tubes, and in each one, 10 grams of protein was placed. Then 20 ml of stomach fluid was added to each of that. So furthermore, 20 ml of stomach fluid was added to each of these test tubes. Okay. Then the test tubes were taken and kept at different temperatures. So they were put in a bath with water or whatever, and they were kept at different temperatures. Now, there's test tube 1, 2, 3, 4, 5. There's 1, 2, 3, 4, 5. Test tube 1 was kept at 5 degrees. Test tube 2 were at 10. Test tube 3 at 20, 35, and 75. Right. So now, let's look at what effect the different temperatures had on the amount of protein that has been digested. So remember that we are now studying the effect of temperature. We are manipulating the temperature. We are changing the temperature, right? And we are looking to measure what is happening or the action of the pepsin. And how are we measuring the action of the pepsin? By looking at the amount of protein that is being digested, because pepsin is a protein digesting enzyme. Right, so let's unpack the data. At 5 degrees, the amount of protein digested was 1 gram. Remember that at a table, you have a unit. So I'm not writing 5 degrees Celsius, 10 degrees Celsius. This degree Celsius means it applies to all the numbers here. Grams applies to all the numbers here. So 1 gram was digested. At 10 degrees Celsius, 2 grams was digested. At 20 degrees, 4 grams. At 35 degrees, 9 grams. So there's a steady increase in temperature. There's an increase in the amount of protein that is being digested. But come 70 degrees, there is 0. Okay? So remember that pepsin is an enzyme. And at 70 degrees, the fact that 0 protein was digested tells us something, okay? That what happened to the enzyme? The enzyme probably became denatured, okay? But we are now talking about the investigation and not the actual process of pepsin. This is just to show you the process of the scientific investigation. But anyway, let's move on. Right, that was our aim of the investigation. This is telling us the method, how the investigation was conducted, and this is telling us our results. Okay, right, now let's look at this. The investigation was carried out to study the effect of temperature on the action of pepsin. There's my test tubes, my temperature, my amount of protein digested. Okay, now we want to identify the variables independent and dependent. We must go to the aim of the investigation. Was carried out to study the effect. This is my aim. Okay, that's my aim. The effect of temperature on the action of pepsin. So what is going to be my independent variable? Did you guess? Okay, I hope you said temperature. Remember, we don't use effect of temperature, influence of temperature. No, it's just temperature. And our dependent variable then is the Action of pepsin. I hope you got that one right. And how was the action of pepsin measured? By the amount of protein that has been digested. Now, remember, we had 10 grams, which we circled, 5 test tubes, 20 mils stomach fluid, and I told you always circle your numbers, 24 hours. Right. Now, state one way in which the reliability of this investigation could be improved. So now, I'm not asking you how was reliability maintained in this investigation. I said, give me one way in which the reliability of this investigation could be improved. Right. So, what speaks to reliability? Remember, we said that how many test tubes were used? We didn't use one test tube. Our sample was 5 test tubes. So how can we improve reliability? We need to look at the number of test tubes that we used, okay? And then how long did we conduct this investigation for? The investigation was conducted not for 1 hour, 2 hours. After 24 hours, we then went and looked at our investigation. So reliability, more than 5 test tubes must be used in order to improve reliability. If I ask you for how was reliability maintained in this investigation, you'll say 5 test tubes were used. But when I want to improve reliability, you must say more than 5 test tubes must be used. So more than 5 must be used. So in other words, you must increase the sample size. But remember, you can't write that. We want a larger sample size. How many test tubes we use? 5. How do we improve reliability? More than 5 test tubes must be used. The second point is we can do the experiment, repeat the experiment, and here we can say that because this experiment was done once, or we can talk about the fact that it was done for 24 hours and we can do it for a longer period of time. Okay, now, how was validity ensured? Now we're going to look at experimental design. What experimental design here was carried out? Okay, we used 10 grams of protein. What kind of protein? Can we use different kinds of protein? No, we must use one kind of protein. And 10 grams of protein was placed in each of the 5 test tubes. I didn't place 10 grams in one, 5 grams in one, 2 grams in one of the test tubes. I placed 10 grams in each of the test tubes. I placed the same amount of stomach fluid, 20 mils, in each of the test tubes. That's how I was trying to maintain validity, okay? So the same amount of protein was used. Yes, 10 grams was used. So here you will say 10 grams of protein was used, which was the same amount in each test tube. And the same amount of stomach fluid, which was 20 mils, was used in each of the test tubes. And then you could say the time was the same for all the test tubes, meaning 24 hours. 24 hours. Now, if I ask you for how can validity be improved? This was how validity was ensured in the investigation. Now you must tell me how it could be improved. So now you look at the investigation, but you don't give me an answer exactly from here. Your answer has to be from your head. Okay? So the pH of the solutions should be kept the same. So in other words, if we are looking at a protein digesting enzyme, they cannot have different pHs. Remember, that will affect the investigation. Okay? The stomach fluid must be from the same organism. You can't take stomach fluid from different organisms. So my point here, and the same type of protein should be used. My point I want to make here is that if you're looking at how validity was ensured, you can find it here from your investigation, okay? But how it could be improved, you must think of something that is not in the investigation. So something that is other than what has been given to you, okay? And that is how you could improve validity. Right, we're going to look at another experiment, another question. Learners from Letzibucho Girls that's in Johannesburg West, Letzibucho Girls School of Specialization. They conducted an investigation to determine the effect of different light intensities on the rate of transpiration in leaves. Right, I'm using simple examples so that you can hopefully understand the concept. So they conducted an investigation to determine, immediately find the aim. That's going to be our aim. Right? The procedure they followed, the following procedure was followed. This is the stem of the question. Read the stem of the question before attempting the question. 2 leafy shoots, 2, underline it, remember it's a number, of the same plant we used. The leafy shoots were of the same age. The leafy shoots were then labeled shoot A and B. Leafy shoot A was exposed to different light intensities, and leafy shoot B was kept in the dark. All other factors affecting the rate of transpiration were kept constant. A potometer was used to measure the rate of transpiration in both groups. The results were recorded every hour for each group. Okay, that's telling us how they conducted the investigation. Now, we're going to look at the question. But before we look at the question, let's look at the data that has been provided. There's a graph. We have rate of transpiration on the y-axis. We have light intensities on the x-axis, 5, 10, 15, 20, 25. My light intensity, my rate of transpiration in milliliters is 1, 2, 3, 4, 5. And there's a steady increase. And then there is a line that is almost a slight, this one here was constant, a slight increase, and then constant again. So we have an idea of what is happening. Right, now we can go to look at the questions. What is the independent variable? Let's go to the aim. Conducted an investigation to determine the effect of different light intensities on the rate of transpiration. So, did you say different light intensities? Right? It was not one light intensity. It was different light intensities. Therefore, we use the word different here. And your dependent variable is the rate of transpiration in leaves. Now, what is the purpose of shoot B? Remember, leafy shoot B was not exposed to different light. It was kept in a dark area. We are looking at, in our investigation, the effect of different light intensities on the rate of transpiration. So I am testing the different light intensities. Now I must remove light intensity. So therefore, B, light intensity was removed because it was a dark area. Therefore, B is my control. I hope you got that right. Shoot B was used as a control. The results of shoot A and B can be compared to those of shoot B. Now, how did the learners ensure validity? We're looking at experimental design. They used the same plant. They didn't use different plants. They used the same plant. So they could measure the rate of transpiration because the plant because it was the same plant, it gave off, the transpiration rate occurred similarly in all the plants because it was the same plant. They used the leafy shoot of the same age. They didn't use a young leafy shoot and an older leafy shoot with more established leaves, for example. Okay, so that's how they ensured validity, right? The leafy shoots from the same plants were used. They were the same age. The results were recorded every hour in the group, and a potometer was used to measure in both the groups. Okay, so remember, whatever is being said here in the stem of the question is not to it's not to confuse you. It's not to add extra information for nothing. Use the information. They're telling you the information here. It's for a reason. They're giving you all this necessary information. Right. Now, how can we improve validity? Remember, we use 2 leafy shoots of the same plant. In order to improve validity, what must we do? We're improving validity here. Remember, we're not talking about reliability. We're talking about validity. So now we are looking at the same plant, the same age that was to ensure validity. How can we improve validity? We have to think of something that is outside the scope of this question, outside what has been given to us. What has not been given to us? The same environmental conditions we used. The same humidity, same wind factors, same temperatures, right? They must take the measurements at the same time of the day. They can't take measurements one in the night, one in the day, one in sunlight, one not in sunlight. Okay, so this is how they can improve validity. So I hope you understand validity speaks to experimental design, and you must know the difference between ensuring validity and improving validity. Okay, now we're looking at reliability. How can they improve reliability? Right? So we are looking now at sample size and the length of the investigation. So they use 2 leafy shoots. In order to improve reliability, we must use more than 2 leafy shoots. So use more than 2 leafy shoots to increase the sample size, and we can do the investigation more than once, because this was one procedure that was followed. We can do it more than once. So do you think that this investigation is reliable? The fact that they did it once? I don't think so, because they've done it once. In order to make sure that this result is reliable, we must do the investigation more than once. So I don't think that was very reliable. What is the conclusion of this investigation? We must go to the results. There's our results in the form of a graph, right? We can talk about each section here. The rate of transpiration increased steadily from the light intensity of 5 lux to 10 lux. And then it was constant from 10 to 15. It improved slightly between 15 and 20, and constant again between 20 and 25. So what can we say? As an increase in light intensity increases the rate of transpiration up to an optimal point where no increase occurred any further. Right. I now want to look at another question. So this is question 2. Actually, don't mind that. It's question 2. Now we are looking at something that pertains. I did a simple example for you earlier that was to show you how to understand the different concepts. But now we're coming to something that affects grade 12 learners, which is the eye. Now, remember to read the stem of the question, number 1, unpack the data, and then go to the questions. In an investigation, Lindy was asked to put a cotton thread through the eye of a needle 10 times. Both eyes were open, and then only one eye, the right eye, was open. So let's look at it again. Lindy was asked to put a cotton thread through the eye of a needle 10 times with both eyes open. And then it was done under the same light intensity at a distance of 50 centimeters. Okay? Right. And then she was asked to do the same thing, but with only the right eye open. Okay? It was done 10 times, 50 centimeters away. Both eyes open, one eye open only, and that was the right eye. Then the results are in the table below. There's a 10 times here, 1 to 10. That was the attempts. There's the amount of time taken to thread the needle in seconds with both eyes open and only the right eye open. Let's unpack the data, guys. In the first attempt, with 2 eyes open, she took 12 seconds. With only the right eye open, she took 38 seconds. That is, now she took a longer time with only the right eye opened. With 2 eyes open, again she took 12 seconds. With 1 eye open, 35. Then 10, 37, 11, 36, 9, 34, 9, 33, 10, 30, 8, 31, 7, 29, 7, 28. As I'm going down here, I can see the time is getting less. As I'm going down here, I can see the time is getting less. But when I compare these two, this one is much faster than this one. So with 2 eyes open, it is faster than with only the right eye open. So I've unpacked the data. I've understood what the table is saying. This is going to help you answer the questions. Apart from the factors mentioned, that means you're not going to talk about anything that was mentioned here in the question. State 2 other factors that had to be kept constant. Think about it, guys. Think about it. This is going to take time in the exam, so make sure you leave adequate time to understand the scientific investigation question. You are threading the needle, right? She's using a thread of cotton. Remember, if I change anything now, I'm going to make this result invalid. Okay, so I'm trying to ask you validity in another way. I'm trying to ask you what other factors must be kept constant. In other words, how can I improve validity of this investigation, right? That cotton thread cannot be a thin cotton thread the first time, and then thick string the next time, thick wool the following time. Maybe she's colorblind. You can't give her red in the first instance and then give her white the next time. It must be the same kind of cotton, the same thickness of the cotton. Okay, so remember it's 2 marks, and state 2 others, we mark the first 2. When you see state 2 in capital letters, we mark the first 2. So give the 2 most important answers first. Right, the size of the needle. Oh, the size of the needle. You can't give her a needle with a huge eye and then a small eye. The size of the needle itself must be the same. The size of the eye of the needle must be the same. The thickness of the thread, the color of the thread, the time between the attempts. So it must be constantly 2 minutes in between each time, so her eye is not too tired. The distance between the needle and the thread. So you're starting here each time, 10 centimeters between the needle and the thread, not one starting here, one starting there. Okay. So can you see this is not given to you in the question? It is other factors that must be kept constant. So this question is asking you validity in a different way. How did we ensure validity? There were 10 attempts. We used a cotton thread. Only the right eye was open for all. Both eyes were open for all to get results. But when we want to state other factors, then you need to talk about what was not given to you here, and that was the size of the needle, the same size eye of the needle, the thickness of the thread, color, and these factors here. State a general conclusion that can be drawn from the results. By you unpacking the data, you already got the conclusion. Remember, we said that with 2 eyes open, she threaded the needle much faster than only when the right eye was open. So you could say it takes more time. Can you see there's a tick to thread a needle with 1 eye open compared to having both eyes open? For 2 marks, or it takes less time to thread the needle with both eyes open compared to having 1 eye open, or the more attempts undertaken to thread the needle takes less time. Any of those could give you your 2 marks. Remember, we are talking about the conclusion, and you have to speak about your dependent and your independent variables, and we are looking at the time, and we are looking at then the attempts. Right, so now give a reason why more than one attempt was made in this investigation. Obviously, that is to improve reliability, okay, because we did the experiment more than once, so we did 10 attempts. How can describe the changes that will take place in the eye if the distance between the needle and the eye was reduced? Now we are coming to the functioning of the eye. The fact that we are looking at the distance, D for distance, what process are you going to talk about here? D for distance, what changes would take place if the distance between the eye and the needle was reduced? So what they did was, they moved the distance between the needle and the eye of the needle from 50 centimeters to 20 centimeters. We are talking about distance. We are reducing distance. Therefore, from 50, we are bringing it to 20. And then we are speaking about near vision. The fact that we're talking about distance, D for distance, the D here, we're going to look at accommodation, right? And the fact that we are moving from far, we are coming now to near, we're going to discuss near vision. And when we discuss near vision, we are going to look at, let me try and get an eraser. The ciliary muscles contract, remember, C for near vision. Okay, firstly, let me say this. When we're speaking about accommodation, it's accommodation because we are speaking about distance. You need to understand that for accommodation, there are two muscles that are involved. Number one is suspensory, and number two is ciliary. And you have to use the word of the iris. So ciliary muscles of the iris contract, that's CC, right? Suspensory ligaments relax. The lens becomes more convex, or you could say tension. The stroke means more, or tension on the lens is released. The sclera is pulled forward. The refractive power of the lens increases, and a clear image forms on the retina. So guys, when you're speaking about accommodation, you need to come up with the muscles, and then you need to know what happens to the muscles, right? The ciliary muscles contract and the suspensory ligaments slacken. And because it's accommodation, D for distance, you're talking about accommodation also has the D. This is just small tips to remind you how to differentiate between pupillary mechanism and accommodation. Right, we're looking at one more question 3, another activity here. Tokozile conducted an investigation to determine the effect of distance on the curvature or the thickness of the lens of the human eye. Distance, okay, we are looking at distance, but let's see. She sat in a well-lit room. She covered her one eye with a patch. A pencil was held in front of her uncovered eye for 10 seconds. She focused on the pencil until a clear image could be seen. At the same time, the curvature of her eye was measured with an optical instrument. The curvature of the eye, the curvature of the lens, okay? The pencil was then moved to different distances from the eye, and it was measured at each time. The results are shown in the table below. The fact that we are talking about distance, guys, distance from the pencil was 10 centimeter, and the curvature of the lens was 4. Then 20 and 3.6 30, and 3.2, 50, 2.9, 100, 2.7, 150, 2.6, 200, 2.6. So this is the same, but you can see here as the distance of the pencil from the eye increases, the curvature of the lens then decreases. And when we're speaking about distance and the curvature of the lens, distance, immediately you need to know that we are speaking about accommodation. There's your D. And the curvature of the lens. I just want you to know, let me just erase that in that way. This is the shape of the lens. The shape of the lens is convex, right? When we're talking about the curvature of the lens, the curvature, you need to state whether it becomes more convex or less convex. This is very important because the shape is convex. Please don't use words like fatter and rounder. That's wrong, okay? You must use the words more convex or less convex. So the results are shown in the table below, and you can see the curvature of the lens was more convex, and then it became less convex. Can you see from 4 millimeters, it went down to 2.6. So we've unpacked the data. Now, what is the dependent variable? So I want to go back. An investigation was conducted to determine the effect of distance on the curvature of the lens. Right. So let's go forward. What is the dependent variable in this investigation? We're asking you dependent one first. Okay, you are measuring then the curvature of the lens of the eye. That's your dependent variable. Your independent variable will be the distance of the pencil. State 2 factors that must be kept constant in this investigation. What do you think must be kept constant in this investigation? Right. It is the amount of light, the time taken to focus on the pencil. You can't use a big fat pencil, a different pencil all the time. And which eye are you using, right? The same eye must be used in both instances, okay? In each of these instances, you must use the same eye. Let's see this. Explain why the factors mentioned in question 2 must be kept constant. So that is in order to increase the validity of the procedure, to get valid results, or to get the results for what is being tested so that the variables do not affect the factor being tested. Okay? We want to get results for what is being tested. That means the curvature of the lens. We want to see the shape of the curvature of the lens with the distance of the pencil from the eye as we're moving it away. So we don't want any other factor to interfere with what we have decided to measure. Describe the relationship between the distance of the pencil from the eye and the curvature of the lens. When you unpacked your data, you already got this information, right? The curvature of the lens of the eye decreases. It decreases as the pencil moves further away from the eye. And further than 150 centimeters away, there is no change in thickness. Remember, both are 2.6. Name 2 structures in the eye that are responsible for the changes. Remember, we spoke about accommodation, and the 2 factors will be ciliary and suspensory ligaments. Okay, it's ciliary and suspensory. Please don't mix it up with the other muscles. Okay. Right, the last paper I want to look at is the DBE paper. This is a May-June supplementary exam, paper 1, 2024. And I've got it here because that's where you can go back and look if you want to see this paper. Scientists conducted an investigation to determine the relationship between age and fertility in men. Okay, they want to determine, that's my aim. This investigation was done from 1999 to 2017. That's approximately 18 years, using 1294 healthy men from the age group between 16 to 65. Men with occupations, working environments involving exposure to excessive heat were excluded from this selection of participants. The following was a procedure. This is the method which you must read as well. Semen was collected after 5 days of no sexual activity. A specialized microscope was used to determine the sperm count or the normal number of normal sperm per milliliter of semen, and progressive motility, the ability of sperm to swim effectively in a straight line. An electron microscope was used to determine the sperm necrosis, that means the immature or dead sperm per fresh semen sample. Remember that sperm is found in semen. Okay, semen is made up of various fluids from the accessory glands. Identify the independent variable. Let's go back. They wanted to conduct the investigation to determine the relationship between age and fertility. Okay, so what did we manipulate? We chose men from a certain age group between 16 and 65. So that means that the independent variable was the age. The dependent variable was the fertility, okay, that we have measured. State 3 ways in which the dependent variable was determined. Let's go back. Right. They determined the fertility in men. And how do they do that? They exposed them to excessive heat. Men were excluded. A specialized microscope was used to determine the sperm count. So that's how they determined the fertility, by using the sperm count. So let's go back. Right. 3 ways in which the dependent variable was determined. They determined the sperm count, the number of normal sperm per milliliter of semen. They worked out the progressive motility, the ability of the sperm to swim effectively in a straight line, and they worked out the sperm necrosis, the immature or dead sperm per fresh semen of sample. Give 2 reasons why the investigation can be considered reliable. Why do you think it was reliable? It was conducted from 1999 to 2017, over 18 years. And then we look at, that was the length of the investigation. And then sample size, 1294 men were tested. So they use a large sample size, but you got to give the number of the sample size that was used, because remember, we are moving away from generic answers. Explain why do you think men with occupations involving exposure to excessive heat were excluded from the investigation? Why do you think so? The age will be the only independent variable. High temperatures affect fertility, right? The sperm count, motility, and sperm necrosis are affected by high temperatures. So that would decrease the validity of the investigation, because now the sperms, the measurement of the sperm won't be correct, because they will be exposed to temperature, to high radiation, and that will kill the sperm, not the age. We want to determine the sperm count or the fertility with regards to age only. But if you are going to include men who are exposed to high temperatures, it's temperature and age that will affect the investigation. So you are affecting validity, you are decreasing validity of the investigation. We want to determine the age. As the man's age increases, how is his fertility changing? And we are doing that by measuring the sperm count. By exposing him to high radiation, the sperm count is going to decrease. So now we don't know whether age is affecting his fertility or the radiation. Therefore, those men were excluded from this investigation. Right. So, learners, I hope that you have understood this section on scientific investigations. The only way you're going to be successful and master this technique is by watching this video again, maybe, and doing examples. Practice, practice, practice makes perfect. Okay, so all the best for your exams, and remember that if you need to read the question carefully, the stem of the question, unpack the data that's given to you in the table or the diagram, find out where's the aim to get your variables, look at validity and reliability and the control. Do not use generic answers, use what is being given to you in the question, and you should be A for away. All the best. Thank you.